Hi everyone. So this session is in continuation with the previous session. Here also we will be summarizing few of the concepts that we have done in the previous lectures. Like I mentioned in the previous session that this would be the last session for this course, but I realized that there are way too many topics to be covered. So I split the last session into two parts. So this will cover few of the topics, and in the remaining lecture we will complete the remaining of the topics. So let's get started. So today we are going to cover these topics, which will cover the immune systems and the digestive systems. So let's start. So let's begin with immune system. So we have already done this, where we have seen that yeah. So we have already done this, where we have seen that the immune system consists or is it can be divided into very many types of immunity, classified into various types. So one of the classifications divide them as innate and adaptive immunity. So innate immunity can be said as the body's first line of defense system, where it means that these are the cells, or these are the immune responses which you are born, and that's why they are rapid in nature. So they are rapid responses, and they mainly consist of the macrophage cells, natural killer cells, the neutrophils, eosinophils, basophils, dendritic cells, T cells, and the natural killer T cells. And then we have adaptive immunity. So this is the immunity that an individual acquires after the birth during his life. So it develops throughout the lives, and that's why it is mainly consisting of those cells which can generate memory. So for example, B cells, which have antibodies, and then memory B cells. And similarly you have the T cells, which are further differentiated into CD4 plus T cell and CD8 plus T cells. Other than that, immunity can also be divided as passive immunity and active immunity. So passive immunity is where the antibodies, which are the main players of the immune system, these antibodies are developed by some other sources, and the person who is receiving it gets directly a pre-made antibody from the outside source. So this, and because it is being delivered via some other source, this immunity lasts for a short time. So, for example, for this will be the antibodies that are present in the mother's breast milk, and it is passed on to the infants. In contrast to this, if there is, we say, active immunity, we mean that the production of antibody in one's own body. So that response is your body's response to a pathogen. So your body is actively producing the antibodies. So these are the classifications for immune system. And then in the active immunity, where your body is producing antibody and also responding to the pathogen, it can be further divided as humoral and cellular immunity. And so both these immunities, because they're part of active immunity and they are part of the adaptive immunity, they have lag period. That means they take time to identify and then come into play, but they are active against wide variety of pathogens. And as I mentioned, that these possess the immunological memories. And here, if we are dividing into humoral and cellular, what we mean is, by humoral we mean something which is via antibody or via a source. So this generally consists of the B cells, which are further differentiated into memory B cell and the plasma cell. These plasma cells are the one that synthesize the antibodies. And for the cellular, as the name suggests, cells. So which cells are primarily involved in this immunity? It is the T cells. And then these T cells are further again classified as CD8 plus and CD4 plus cells. And why are these different type? Because they recognize different type of antigens and response accordingly. And once they bind to the antigen, they are able to recruit more T cells, which further elicit the immune response, and then help in the disruption of the pathogenic cells. Okay, so we will go into detail about how this actually happens. So let's start with the humoral, which is the antibody mediated immune system. So here what happens is yeah. So here what happens is, there is a free circulating pathogen in the system, and there's a B cell that binds to this antigen, and this B cell is specific to the antigen. And then there's a T dependent B cell cooperation with the TH cells. So TH cells are T helper cells, and T helper cells are these cells. T cells, TC cells. They are also called TH cells, T helper cells. So these T helper cells are recruited via the CD4 plus T cells. So you have these cells coming into play. So these B cells, they recognize the antigen, bind to them, and then they proliferate. And from the proliferating progeny, one of them will become, or some of them will become plasma cells, and then some of them will become the memory cell. So then, if it's a plasma cell, it will then lead to production of antibody against the antigen based on the specific antigenic sequence that is bound to the B cell. And if it is memory cell, these memory cells, they are, so what happens is these retain the memory of the antigen. So the next time the same antigen attacks the body, the response is very quick because the antigen is already recognized by the body. These memory B cells respond very quickly to the second encounter of the pathogen. And if we're talking about cellular or cell mediated immune system, what happens there is, so when there is an exposure to antigen, which is generally of the intracellular nature, let's say a cell, for example, which is infected by virus and bacterium and parasite, so these enter the cell and they are presented on the cell. What we mean by presented on the cell is they are, because they're intracellular, they come to the surface of the cell and they mark these cells. So these, what happens after this, a T cell will then bind to this via the MHC antigen complex. So MHC is a major histocompatibility complex. We'll go back here and we will see this. So there are some cells like this which have this MHC class 1 and MHC class 2. So these MHCs, they recognize antigen and they present it on the cell surface so that these T cells can come and bind to them. So these MHC antigen complexes are bound to by the T cells, and these are on the surface of the infected cells. Once the T cell binds to this antigen MHC complex, they get activated, and what happens after there is activation of macrophage. So this T cells then further recruit the macrophages, and we know that macrophage are able to do phagocytosis. So they kill these infected cells, and subsequently you will have the CD8 T cells, which are the cytotoxic T lymphocyte. Basically, these are able to induce apoptosis. The name says cytotoxic. Basically, these means cytotoxic, so toxic for the cells. They will induce apoptosis of this target cell, and they will lyse the target cell, and the contents of the cells are exposed in the as debris. So this is how generally they work. So the idea to remember here is humoral means antibody. Antibody will have a specific sequence to bind to the antigen. While for the cellular, which is a T cell mediated, there you will have the intracellular antigen, which are presented on these infected cells surface, which are recognized by the major histocompatibility complex called as MHC class 1 or class 2. How do they differentiate? They differentiate on the basis of the cells that they recognize for binding to. And then once the MHC class 1 and class 2 are bound to the antigen, these will further bind to the T cells respectively, whichever they bind to. So it is again coming back to here, if it is MHC class 1, they will bind to CD8 plus T cell. If they are MHC class 2, they will bind to CD4 T plus cell. If it is CD8 plus T plus cells, if it is CD8, it is a cytotoxic T lymphocyte. So this will directly kill the infected cells. However, if it is CD4 T cell, then it is the TH cells. So these are TH cells. So what will TH cells do? They will further recruit several components that will elicit the immune response. For example, they will recruit interferons, interleukins. All these are inflammatory responses cells, and they will also recruit the B cells. These B cells will further again. So this is T cell dependent B cell response. So here again the B cell binds to it, and then it starts producing the antibodies. So you see how these two function together. Okay, so moving on, let's now discuss the antibodies, which are also very important to discuss. So antibodies, as you can see, they are this Y-shaped structure. They are made up of 4 polypeptide subunits, and they are each consist of these 2 identical light chains and heavy chains. So these are the heavy chains, and then these are bound by the light chains. In the light chain and the heavy chain, you have two main division: variable region and the conserved region. For the light chain, this is called the VL region. For the in the light chain, the conserved region is called the CL. Whereas for the heavy chain, the variable portion is called the VH, while the conserved portion is again divided into several parts. For example, CH1, 2, 3, and this is exactly same for the other other UFM unit as well. Now, these two chains are connected to each other via disulfide linkages and this hinge here, which is also rich in proline residues. Now, in the antibody, you have these regions here that binds to the antibody, antigen, sorry. And these are the N-terminal region. And here, because they bind to the antigen, they are called the fragment antigen binding domains. So these are antigen binding domains. They are called the FAB domains. Similarly, here you have the C terminus, which are called as the FC. So fragment crystallization domain. And what they do is they interact with the effector cells. So if the antigen binds there, these bind to the effector cells, they are able to induce the apoptosis of the antigen or the phagocytosis of the antigen. And so what you see, what is the importance of variable and conserved region is that, so as you see, these are conserved region and they were binding to the effector cells. So conserved means that these portion or these fragments are similar, or they have the same sequence all across the antibodies, because they are supposed to bind to the effector cells, so they have to be constant. But here, the variable region is actually the part that binds to the antigen. And antigen can be of diverse types, and they will have diverse sequences. So these are variable region and they keep changing. And the change is important because that changes the sequence of these region, which is called the paratope. So this paratope region will keep changing, and that will enable it to bind to different antigens. Otherwise, if it is also constant, or if it has a only limited repertoire of sequence, then it will bind to only certain antigens, and that is not what the immune system desires for. The immune system wants to bind to and neutralize most of the antigens. So that for that reason, this variable part is very, very important. And like I mentioned, this part which binds to the antigen, it is also called paratope. And the antigen part which binds to the antibody is called the epitope. The epitope is the region that is recognized by the antibodies. And there are 5 main types of antibodies, which are IgA, M, G, E, and D. And all of these have their own characteristics. For example, you have IgA, which is like the first antibody to be passed on to the infants by the mother's mother. And you also have IgE, which is involved in the inflammatory responses. And then you have several others which are again different in their structure, different in their regions that they bind to, different in the kind of responses that they elicit. Now these antibodies, as we have already mentioned, that they are synthesized by the B lymphocytes, but we also saw that this kind of responses, there is a lag period. So we'll see why is that lag period there. So what happens is, whenever an organism's immune system encounters a foreign particle for the first time, when it is the first time and there is no memory cell to recognize it, the first response is the macrophages come, they interfere and they capture and they break the cells. This information is then passed to the B cell, as we have seen that they are recognized by these MHCs, and then they pass information to B cell. Now, once these antigens they are presented to the B cells will bind to them, will begin the production of the new antibody based on the specific epitope that the paratope is bound to. Epitope being the region of the antigen. So the antigenic region is bound to the B cell, and the B cell will now start producing antibody. Now, after it has begun to produce the antibody. So now we have entered this phase where it has started to produce it. Now, each lymphocyte of B cells will generate a unique antibody against a unique epitope. That is why it is also important to know that not every B cell will bind to the same epitopic region. Different B cells will bind to different epitopic region, and that will generate specific antibodies. So you have this one antigen being bound to by different antibodies at different sites. Then once this is done, once the B lymphocyte has been synthesized, these B cells release the antibodies, which then go and bind to the specific pathogen, and they result in the elimination of the pathogen from our bodies. How does that happen? As we have already seen, it can happen via direct mode where the antibody directly goes and binds to the pathogen, or it binds to the pathogen surface and then recruits several other components from the immune system to eliminate the pathogen. And then there are some memory cells which are remaining in the body, which respond to the secondary attack by the antigen in a faster response. So when you have the production of antibody, you have this lag phase in the initial part where it is still trying to first being presented to the B cell. Then the B cell starts to produce the antibodies. It reaches a plateau where it has started neutralizing and binding to most of the antigen, and then there is a decline in the synthesis or the production of the antibodies. This is what exactly we see here. Here, there are some things that I will like to highlight. So again, we'll move from here. So once the B cells have activated and they are proliferating, and then they differentiate into different kind of cells, you have then first the antibody secretion. Then you have isotype switching. So isotypes means that in the some antibodies, you have this conserved region that can switch among themselves so that they can generate diversity in the antibody. They have affinity maturation. So what happens is when these B cells and the antibodies, they bind to the antigen, they start maturing at a faster rate, and then based on that binding region, they modify their paratopic region, and then you have the memory B cells. So these are the different kind of antibodies outcome that can happen after the B cell is activated. Now, antibodies, because I mentioned that they bind to the antigen and they are specific, they can be all different types as well. They can be monoclonal or they can be polyclonal. So if they're monoclonal, which is in this case, as you can see, so the antibody can bind to like one region of the antigen, or they can only bind to one antigen. But you have polyclonal. So polyclonal means there are several the antibodies that can bind to different parts of the antigen. So the antigen can be bound by different antibodies. So generally, polyclonal is what what generally is produced in general immunizations. Example for example, if you would have heard about vaccinations where you immunize the horses with certain antigens, and then the horses are bled, and then from the blood the antibody is purified, that is used as the commercial antibody or vaccine. So those are generally polyclonal because the antigen which is present, it will have several surface receptors, and then antibodies are secreted or synthesized for different epitopic regions. So that's why they are generally polyclonal, and this happens mostly in vivo. And what other factors are related to this is, they, from a production point of view, they take very short period of time, and they are very easy and cheap to make, and they have very large variability because they are making they are recognizing different parts of the antigen, and they can show cross reactivity. When we see cross reactivity, or when we say cross reactivity, what we mean is, let's say the antibody is generated against species A or a specimen A, but it is also able to neutralize or react with the specimen B, and that is based on, let's say, some kind of homology between the specimen A and B, or the epitopic region being the same. So there is cross reactivity because there is high variability. Now, in contrast to this, you have the in vitro, which is in vitro production of antibody, which is called the monoclonal antibody. As the name suggests, it is made only against one epitope, and that's why it's very expensive, because you need to first get the specific epitope and then generate the antibody against it. And then, of course, because it is, you have to make it against a single epitope, it takes a longer time to synthesize. However, you can scale it up to a high extent because you are doing it at an in vitro scale, like in a ex situ condition. It's not in some natural animal. You are doing it in a cell environment, lab environment. And so the scaling up is quite high, and the chances of cross reactivity for these antibodies are very low, because obviously they are generated against one very single specific epitope. So the technique by which monoclonal antibody are generally produced are called the hybridoma technology. Okay. So again, I mentioned in the previous session as well, that There would be some topics that you would like to know about that topic more. For example, if you wanted to know about hybridoma technology more, I've given the reference to the actual session where we have discussed these topics in detail. So you can go back to those PPTs and you can see them. Okay, moving on. So this interaction between antigen and antibody, they are of different types. What I mean is this is by this is that in a setup of and clinical environment, you can detect this antigen antibody reaction by several kind of reactions like precipitation, agglutination, complement fixation, or immunofluorescence, or ELISA. All these will help us to know if there is an antigen antibody interaction. And where do you use this in a clinical setup? Generally to identify if there is a certain antigen present. For example, if there is a certain virus present, or if there is certain kind of antigen which is present in the body, or for example blood test. So I'll talk about two of these kind of reaction, precipitation reaction and agglutination reaction, and how they are used for the clinical setup. So in precipitation reaction, you have an insoluble precipitate forming for the antigen antibody complex. When you have a soluble antigen and you produce the antibody, and they are combined in the presence of some salt and a specific pH and temperature. And here you are anting the antibody to understand if that specific antigen is present or not in the cell. If you see a precipitate, it means that there is an antigen antibody interaction, which means that whatever you are testing for in the patient, the antigen is present. And the other one is agglutination reaction. So here you have, instead of these insoluble precipitate, you will have some particles clustering of agglutination in the presence of antigen antibody, and those clumps that you observe which of the cellular antigen will let you know that if such antigen is present in the patient or not. One such example, or one such example where it is widely used, is the blood group typing. So blood groups A or B, they are classified on the basis of the surface antigen that is present on the red blood cells. So A means a certain kind of antigen, B means a certain kind of antigen. Both the antigens present together give you an AB blood group, and none of the antigen on the surface gives you an O blood group. And the positive or negative refers to another antigen, which is a rhesus factor. So that presence of that factor makes you positive, and the absence makes you negative. So if it is an AB positive, it means you have all the antigens present, and if you're an O negative, you have none of the antigen present. Now, how do you test it? So you take these kind of tube, you add the blood as one droplet, and then on each column here you see the antibodies being added. The anti-A means antibody to A. So antibody to antigen A, antibody to antigen B, antibody to the D or the rhesus factor, and control. Now, wherever you see agglutination, it means that antibody is, or that antigen is present, and the antibody is forming an agglutination with this antigen. So if you're an O positive, ideally you should have only one factor present, which is the D factor. Because it is O, that means there is no anti-A and anti-B, but you see clumping in the anti-D antibodies, and that's why this gives you a phenotype of O positive. In O negative, you should not have this clumping as well, because even in the negative, you don't have the rhesus factor or the D antigen as well. Now, if you're going to compare this to AB positive, you will see clumping in all the three wells. Control is where you don't add any antibody. So you should not see any agglutination whatsoever. If you see any such cases, especially in the control cases where you also see agglutination, then you know that whatever test you have done here has got some problem. So that's why it's an invalid. Rest of the cases, you see that wherever the antigen is present, you see an agglutination. Wherever it is not present, you don't see an agglutination reaction, and that gives you an idea of the blood group of the patient. Okay. Now we have spoken about the phagocytosis, which is induced by the antibody. So we'll try to understand how does the phagocytosis actually takes place. So as you can see in the animation, so this is phagocytosis, where you have an antigen being taken inside the cell, fusing with the lysosome and getting disrupted via the hydrolytic enzymes present in the lysosome. So you can see this step by step here. So this phagocytosis process is generally carried out by cells like neutrophils and macrophages. And what do they do is that these cells, they try to destroy these foreign particles or pathogens, for example, bacteria or any infected cells, by engulfing them and then lysing them with the lytic enzyme present in the lysosome. So they are taken inside as a vesicle via these receptors on the surface, and once they are engulfed inside, they are, this vesicle is called phagosome. These phagosomes will go and fuse with the lysosome, as you can see here. These are then called the phagolysosomes. Here the lysosomes hydrolytic enzymes will come into action, and they will lyse this cell, and this cell and the debris are then exuded by the exocytosis process. So you can see here how exactly it takes place. So you have this chemotaxis. Chemotaxis means how the cell actually like recognizes the microbe. Then the ingestion, then the formation of phagosome. You have the fusion of the phagosome with lysosome, which is called the phagolysosome. Then you have the digestion of the ingested microbes by enzymes. Formation of the residual body containing indigestible materials. So these are the debris, and then the discharge of the waste materials. And the once, so the main thing is here in these phagolysosomes, the activity of the enzymes get enhanced because of the decrease in pH, and when these are acidic environment leads to the activation of the enzymes in the lysosome, which result in the disruption of these cellular particles. Okay, so based on this discussion of immune system, these are some questions that I have compiled, so I'll go over each of them. Well, I'll give you 10 seconds to answer first. So let's go over it. The first question is, which of the following cells is involved in cell-mediated immunity? It should be easy if you just think what we had discussed. We had discussed several kind of classifications. So this is one of those classifications. Second is, synthesis of antibody takes place by which of the following cells? Where are they actually synthesized? Next, which of the statement is true about passive immunity? And then antigen binding sites are present in which part of the antibody? Yeah, so the first one is cell mediated was T cell because humoral immunity is by B cells. Which of the cells take part in synthesis of antibody? It is B cells. For passive immunity, the following statements are true, which means all of the above, because the first one which says it causes reaction is correct. Second says it develops immediately, that is also correct because it is being passed on as a formed product already, and it's a short period, lasts for shorter time only. That's why the answer is all of the correct, all of them. And this we have discussed that the antigen binding site is present in the Fab region, the fragment antigen binding region of the antibody. Next, neutrophils, basophils, lymphocytes, eosinophils, and monocytes are examples of which of these kind of barriers. Which of these immune cells are able to quickly respond post any subsequent encounter? So the word is subsequent, so you should take a cue from that word. Next, which of the following cells of the immune system do not perform phagocytosis? And which are the following stages? For the following are stages of phagocytosis. So the first answer is cellular barrier because physical barrier and physiological barriers, they are different. Physical barriers are the skin, physiological barriers are the mucous membrane and the gastric acid. Cytokine barriers are the cytokines, so they are interferons, interleukins. So cellulars are these cells which are involved in the defense against pathogen, and which are responsive cells are responsible for the for the subsequent encounter, memory cells, because they retain the memory of the antigen. Which of these do not perform phagocytosis? It is the basophil. And in phagocytosis, you have the pathogen membrane sacs combining with lysosome, which ultimately leads to the formation of phagolysosome. Okay. Next, we will talk about organelles which are involved in vesicular trafficking. So, first, by vesicular trafficking, we mean that the products or the movement in the cells from one organelle to other organelle is mediated by these organelles that we will discuss. And these are vesicular, means the transport from one part to the other is in vesicles. So, the vesicles are passed on from one organelle to the other till it reaches its destination. And in the vesicles, you will have all the products like proteins or lipids which are supposed to be sorted at different locations, some within the cell, some on the cell, some outside the cell. On the cell, I meant at the surface of the cell. So these proteins are targeted at different regions, and these organelles that we will discuss, they are all involved in the completion of the during the process of the protein synthesis or any biomolecule synthesis. So the first organelle that we will discuss is the endoplasmic reticulum, and this, you can see that this is the nucleus of the cell. So these are the nuclear envelope with the nuclear pores, and the ribosomes, or sorry, the endoplasmic reticulum runs in continuation with the nuclear envelope. Now in these endoplasmic reticulum, you have some cisterns. So these are cisterns, these are stack-like structure. Now on some of these stacks, you have the ribosome present. Now these ribosomes give the appearance of a very rigid, rough structure, because of which this part is called a rough endoplasmic reticulum. Well, the surfaces which do not have this ribosomes give a very smooth, shiny like look. So that's why it's called the smooth endoplasmic reticulum. And both of these, because of the presence of and the or the absence of ribosome, they have very different function. For example, smooth endoplasmic reticulum, they are generally involved in the synthesis of lipids or steroids, or they are also involved in the metabolism of carbohydrates, detoxification in the liver, and storage and release of calcium ions. So why? Because these are these have calcium storage pumps, so they can also regulate the movement of calcium, intracellular calcium transport. While on the other hand, the rough endoplasmic reticulum, they are associated with protein synthesis, protein sorting, and as I've already mentioned, the proteins can be sorted in three different manners. It can be an integral membrane, it could be a secretory protein which can go to different regions, or it could also be different organelles within the cell, so it can move intracellular to different regions. The next organelle which is involved in vesicular trafficking is Golgi bodies. So again, these are also these stack-like structure. They also have these multiple cisterns, and they have this lumen inside. So the incoming transport vesicle binds to the cis phase, and the vesicle then moves from one cistern to the other till it moves to the from the transphase to the other side. So from the transphase, the secretory vesicles are moving outside. So they keep fusing, forming a vesicle, and then come out as secretory vesicle from the transphase. So in the generally, the structure will have these kind of 5 to 8 cup-shaped cisterns, and these are all connected by tubular connections. The cis phase generally receives the vesicles from the endoplasmic reticulum. So the endoplasmic reticulum passes these vesicles to the Golgi bodies, and generally at the meat. So the region that actually so what happens in the Golgi bodies after these vesicles are transported or transferred via the endoplasmic reticulum, the protein content is modified. So the protein undergoes modification, so that so the idea is, for protein to go a certain region, there has to be a signal peptide that says that okay, this signal means it has to go to liver, this signal means it has to go to mitochondria. So those modifications were to give it the proper signaling nature, or the signal for its transport is added in the Golgi bodies. So that's what we mean by covalently modified by the sugar. So the addition of several sugars, you have these modification of the protein synthesized. And then the trans Golgi is where you have the Golgi. This part is facing the plasma membrane, so that this side sorts the vesicle for the distinct organelles of plasma membrane. And as I already mentioned, that there are several functions for Golgi bodies. For example, protein sorting, protein modification via glycosylation, or proteolysis. So what happens is, it's not always this anterograde movement. You also have retrograde movement, where you have, let's say, let's say one of the vesicles have some protein which are degraded or not properly formed. So what happens is, instead of sending it forward to plasma membrane, Golgi body sends it backward to this space to either properly fold the protein or just directly disrupt the protein synthesis, which is by proteolysis. So it leads to the lysis of the protein, and that is carried by lysosome if it has to kill or it has to degrade protein. So that's why generally lysosomes are called suicidal bats, because they are involved in autophagy, which means that they operate to starve, like operate to kill the cells, kill the own cell, kill its own cell. And that's because of the presence of this acidic nature, because of which, because of low pH, and they have these proteolytic enzymes and hydrolytic enzyme which degrade the ingested material. And as I have mentioned, what all they can possibly degrade, they can degrade the old protein, pathogenic bacteria, or even the ingested food material which are for delivery through the vesicular system. Now, as we have already mentioned this little bit in the previous slide, that you have glycosylation, which targets the cell for its different destination. And this is one of the most common post-translational modification of proteins. So post-translational means after the protein synthesis, these modifications are added so that the proteins are targeted to their destined location, and these become glycoproteins because they are glycosylated. So they are added, the sugar are added to these proteins, so they are called glycoproteins. And these are of different importance. For example, they are involved in the production of enzymes, they help in cell attachments, they are present on the cell surface cell surface receptor. And how are these sugar added to the protein are also of two types. They are either O-linked or N-linked. What we mean by O-linked or N-linked is basically how and which amino acid or which sugar is added. So, for example, here we have N-glycosylation, which is here N-glycans. So you can see that the constitution is different based on the asparagine sugar on the asparagine amino acid. But for the O-glycans, they are generally the serine or threonine. Remember, these are the amino acid with the OH group, and the glycans are present on these functional group for these protein. And so N-glycosylation, as I have already mentioned, they are talking about these are the glycans that are added to the asparagine residues, that too on the carboxyamido nitrogen. So if you see the structure of asparagine, the N, which is on the functional group of the R, on that nitrogen these locos are added, and via several steps. So you have first the precursor glycan assembly. So these glycans are first like you can say, they're assembled on the membrane of the endoplasmic reticulum or Golgi bodies, and then they are attached onto the asparagine residues. Then they are further trimmed and matured so that they are properly targeted to their destination. Yeah. And for O-glycosylation, as we saw that these are very, and these are mainly common for the high molecular weight protein, and generally in the mucus secretion or the antibodies. And here you have the N-acetyl galactosamine, which are transferred to the beta OH, the OH group of the serine or threonine. And generally the O kind of glycosylation, they form less complex structure because they don't have this precursor assembly. This, when you, we have discussed it in this problem solving session 6, where you go back and check that the N-glycosylation, the whole step of assembling, trimming is very complex, which makes the N-glycosylated amino acids and the proteins very complex in structure. Okay, a brief questions and answer for this topic. So just based on what we have discussed so far, the first is, which are the following statement is true about Golgi bodies? Second is, where does the ribosome and polypeptide chain complex get targeted? Was a short segment of polypeptide chain is synthesized during protein translocation. Next, the transfer vesicle from RER fused with which region of the Golgi complex? And the next is, what is involved in the synthesis of phospholipids? So, given these different organisms, which one is involved for the phospholipid synthesis? Yeah, so the first one is all of these are true for Golgi bodies. So they are, it has a sac-like organelle. It is located near the nucleus and it helps in transporting the particles throughout the cell. Next, after the synthesis of the polypeptide chain, they are targeted to the endoplasmic reticulum because they have to move out from the nucleus. And as we have discussed this already, from the rough endoplasmic reticulum, if there is a transfer vesicle, or basically from the endoplasmic reticulum, if a transfer vesicle is coming, it fuses at the cis terminal of the Golgi complex. And if there is a synthesis of phospholipids, it is generally in the smooth endoplasmic reticulum. Okay. Next is, which of the following cell organelles is involved in the breakdown of organic matter? We've discussed part of this. What are the functions of lysosomes? Protein glycosylation refers to addition of which of these biomolecule on what molecule, and what is the function of the glycosylation? So the first one, as we had discussed, breakdown of organic matter is done by lysosomes. Second is lysosomes are also involved in the autolysis, autophagy, and digestion. That's why the answer is all of the above. Glycosylation refers to the addition of carbohydrates to proteins, and glycosylation helps in conferring stability in the protein. So that's why they are added so that they give a structural stability to the proteins. Okay. Now, moving on from this topic, we further move to nutrition and digestive system. So, we first discuss about the different type of nutrition modes that are present. So, first is autotrophic, where you have these organisms who synthesize their own food by using simple inorganic matters like water and carbon dioxide, and using resources from light and the presence of chlorophyll to synthesize their own food in the form of sugars and give out oxygen. This generally happens in plants, in algae, and the bacteria, which are the cyanobacteria. And if you have to classify, or if you just have to outline these steps involved in autotrophic nutrition, you first have the absorption of light from the sun via the chlorophyll. With this trapping of the light energy, then there is a conversion of this light energy to chemical energy via the splitting of water, and which releases the oxygen and the hydrogen. And then finally there is reduction, in which carbon dioxide is getting reduced. So they are using up carbon dioxide, which gets reduced to give rise to carbohydrates, and the oxygen that was generated is given out as the respiratory product. The other mode of nutrition is heterotrophic, where they don't synthesize their own food, rather they use or they depend on the autotrophs for their nutrition. And this generally are of different kinds, what kind of food they are feeding and what mode of food they are eating, like what mode they are using to eat for food. And the first classification is like carnivore, herbivore, and omnivore. So herbivores are the ones that feed exclusively on plants. Carnivores, they feed on plants as well as animals, so they have meats and the omnivore has plant and meat, while the carnivore feed exclusively on the meat. So you, for example, of the herbivores are the ruminants, cows and buffaloes. For carnivores, you have several of the mammals like lion, crocodile, tiger, while for omnivores, you have humans and deer and the hippopotamus, for example. Yeah. Now, these, as I mentioned, based on what mode of nutrition they are having, they can be further divided into three types: parasites, saprophytes, and holozoites. So, parasites means they require food, but from the body of another organism. Again, where do they live, or how do they survive? Also classifies them as either internal, which means that they have to be inside the host material, host body, and only then they can survive. And external means they do not have to necessarily enter the host body, but they can just feed by just sticking onto the surface of the host organism. For example, fleas and insects. Saprophytes, for example, these are those that feed on dead material. So what they will do is they will secrete digestive enzyme outside, digest the food completely, and then swallow these food enzymes. For example, mushrooms or houseflies or spider. These all have enzymes which in their saliva or in their secretions, which digest the food outside and then they ingest that digested food. While holozoic nutrition is where you have the entire food being taken inside and then the digestion is carried out inside the organism. For example, humans or dogs. So they have the food and these food are digested inside the inside the body. So we now move on to digestive system, which generally refers to the conversion of the food that we have taken in into the energy. To study digestive structure system, you can divide them into two parts. First, just by looking at the structure, basically looking at what all components are involved in the system, and then what are the processes that are involved in the digestion process. So structurally, it starts from the mouth, so it has paddings, then it has this long tube, esophagus, then it consists of stomach, then there is large intestine, small intestine and large intestine, and which ultimately leads to the rectum and the accessory organs. While for the process, you have the ingestion, mixing and movement of the food here, secretions of several enzymes all the way in the stomach, from the esophagus, stomach till the intestines, digestion of the food here, conversion, basically conversion of the food material into several simple compounds, and then absorption of the required nutrients, and then excretion of the ones that are not required by the body. So moving into details, I'll try to cover as much as possible, but it's a vast topic, so you can always go back to the problem solving session 7 to actually see, because what I have just summarized them here, but all of these are need to be looked into each individually, so you can go back and always check them. So for example, food, mouth. So it starts from the mouth. So mouth generally consists of salivary glands, teeth, tongue, and all of these what help in the chewing. So food that is entering the alimentary canal happens at this phase, at the mouth stage, and where it is chewed by teeth and broken into smaller pieces by the help of teeth and the enzymes that and the saliva. And then you have the tongue, which helps it in swallowing. Then after that, the food is then passed on to esophagus. So esophagus can be said as a muscular tube, where the food is pushed down to the stomach via the muscular movement called peristalsis. It is then further moved to the stomach. In the stomach, you have the gastric juices, and these gastric juices have low pH, so they are acidic, and because of being acidic, they help in digestion of the food, and they also potentially kill harmful bacteria, because bacteria lies in the acidic environment. But to protect the stomach from these highly enzymatic or acidic condition, you have the lining of these stomach by mucus that protects the stomach against its own acidic secretions. Subsequently, also have these liver and gallbladder and pancreas. These secrete enzymes that are released into the stomach and the intestine that help in the digestion of the food material. So, for example, liver, these will produce bile, and this bile will help in digestion of fats and oils. Similarly, gallbladder is associated with liver because when the bile is secreted, it is stored in the gallbladder, and when it is required, it is released or it is expelled from the gallbladder. And pancreas, similarly, they have several enzymes. So they generally will see that there are multiple of hydrolases, lipases, which are present in the pancreatic juice, all the enzymes, which are also involved in the digestion step. So then from the stomach, the food is passed to the small intestine. Here again, you have the digestive enzymes and the bile, which are acting upon the some of the digested material and the undigested material, and used the food are then converted into simpler products, and here the nutrients are absorbed via bloodstream because of the presence of villi that we will see in the coming slides. And then the food is passed to large intestine, which is mainly made up of two parts, colon and rectum. So this main part is colon, and at the end there is rectum. In the colon, you have the absorption of water, and so basically you have the water reabsorption, which is important for the maintaining of the hemostasis of the body, and the remaining material which is not required by the body is passed on further to the rectum and the feces, and as for the excretion. Okay, so based on these first few topics, there's some questions. So first is the type of nutrition where organisms take in the food that is digested by enzymes secreted outside. So again, which mode uses food which are secreted outside the body? Okay, which is the valve that is that runs between stomach and esophagus? You can try to answer this question. I will come back to it, and I'll try to explain this topic if, in case you have answered it wrong. The next is the gastric glands, which are situated in the thin layer of stomach. So which layer of the stomach actually has the gastric glands? And in general, what is the mechanism of digestion? Like, what are the steps in which what flow? Yeah. So, like I said, if your food is being digested outside only before ingesting, then it is saprophytic nutrition. I'll come to this question. The gastric glands are situated in the mucosa layer. The proper answer or the proper flow of the digestion procedures are ingestion of the food, breakdown, followed by absorption and the removal of waste. So the valve which is present between stomach and esophagus is cardiac, because pyloric is from the stomach to the intestine. Ileocecal, as the name says, it is ileum, so it is in the intestine, and gastric valve is in the gastric. Now, the importance of a sphincter and valve is that they prevent the backflow of the food. So, if the food is passing from stomach to intestine, these sphincters and valves will prevent the backflow from intestine to stomach, so that there is a unidirectional flow of the food in the digestive system. Okay. So the next part is about secretory glands. So all the glands which are involved in the secretion of enzymatic substances in the digestive processes. First, we have salivary glands, which are situated in the mouth region. There are again three type of glands here: parotid gland, sublingual, which is right beneath the like the tongue region, and then the mandible, submandibular region, right below the sublingual gland. So these parotid glands, they generally secrete the saliva, and the saliva, as you can see, that they, as you know, that they are generally help in the mastication. Mastication is the lubrication of the food material and then swallowing of the food material and movement of the food material. The submandibular glands, they are embedded again in, as you can see, in the mucous membrane of the buccal cavity, and they also secrete various sera, fluid, and mucus. So again, these are all contributing to make the buccal cavity amenable to digestion of food. And sublingual gland also, they secrete the mucinous fluid that help in lubrication, and then these aid for swallowing and initial digestion. Moving on from salivary glands, you have the esophagus. So the esophageal glands also secrete mucus, because the main function of esophagus is to pass down the food via peristalsis. So they secrete mucus that enable lubrication during swallowing. The next glands are gastric glands. So these glands are present in the stomach, generally in the mucosal layers. So if you see here, these are the gastric layers. You have the mucus. So these, if you see it on the domains, I'll just go back once here. So if you see this structure here, you can imagine the mucosa here, and then so region that we just saw was mucosa, and the layer beneath it was the submucosa. So in the mucosa, you have, if you see, there are these pits kind of structure which run this longitudinally, and if you enlarge them, you can see that they have these parietal cells, different cells on the surface, each secreting different kind of product. So for example, the parietal cell, they generally are releasing the gastric acid, the HCl, and the intrinsic factor for the calcium 2 plus absorption. Then you have this chief cells. These chief cells which are present here, they are synthesizing these pepsin and gastric lipases. So these names like lipase, they give you the idea of what kind of product or what kind of food they will be acting upon. So lipase will act upon lipids, so they will digest the lipids. Then you have the mucus cell, as the name suggests, that they will be secreting the mucus and the bicarbonate. All these bicarbonate and HCl, they make the environment of the gastric gland, gastric stomach very acidic. And then you also have two different kind of cell. You have endocrine cells, which are the G cells. Generally they produce the gastrin. These gastrins, they trigger the secretion of enzymes and HCl. So generally, if you when you feel hungry, it's because of the secretion of enzymes and these HCs, which is when the acidic content of your stomach goes up, you start feeling hungry, and if you don't eat on time or don't eat properly, this acid then start having refluxes. So you have the acid reflux, and acidity is what you call it. The other is this argentaphin cell or the D cells that secrete serotonin and somatostatin. So these, so as you can see, there's a self-regulation in stomach. So G cell, they secrete gastrin, which stimulate the acid secretion. D cell, they synthesize somatostatins, will inhibit the secretion of acid. So there is a regulation. Nothing goes. So there's a checkpoint within the organ itself. Next, as we have spoken about liver, so your liver generally secretes bile that involves itself in the making of chyme, which is the digestion of fat, the digested material of the fat, and that is generally carried out by bile. And the digestion of fat is called emulsification. Other than that, bile is also responsible for removing excretory products like bilirubin and bile pigments. Next, we have spoken about this as pancreas. So pancreas are also involved in the secretion of several juices that aid in digestion. Along with it, it also produces hormones, which are also related to digestion process by secretion of insulin and glucagon, which is responsible for either the breakdown of glucose or the buildup of glucose, and that it does by the acinine. So here in the pancreatic environments, pancreas, it consists of multiple cells that each secrete different digestive enzymes, which is generally called the islets of Langerhans. And these pancreas, they have connected, so this they have duct, and this duct connects to the hepatic duct, hepatic being the liver duct, and these together empty into the duodenum. The duodenum is a part of this intestine. And as I was mentioning, so in the juices or the enzymes that are secreted by pancreas, they consist of the sodium bicarbonate, several proenzymes like trypsinogen, chymotrypsinogen, and procarboxypeptidases, and enzymes like pancreatic amylase, pancreatic lipase, and nucleases. So you can see that these are different enzymes that function or that act on different biomolecules. Now there are several proenzymes like trypsinogen and chymotrypsinogen. So what do these mean? These mean that when they are synthesized by the pancreas, they are in the inactive form. They get activated only under certain conditions, which is when there is, let's say, there is a requirement to the to digest trypsin. Then this trypsinogen will get activated to trypsin, and that will act on the proteins to digest the protein. Similarly, chymotrypsinogen will also be activated by certain compounds, which will convert it to chymotrypsin, which again will act on proteins for digesting the proteins. And like I was mentioning, that the pancreas has these different hormones, insulin, and then you have the glucagon. So this insulin and glucagon, they are maintaining the level of glucose in the blood. Intestine also have several glands. For example, crypts of Leberkuhn and Brunner's gland, and both these glands, they secrete again several digestive enzymes that help in the digestion of the food material that are passed from the stomach. Now, as I was mentioning, let's see the enzymes that are synthesized. So we have salivary amylase and lysozymes, which is at the buccal cavity. So as the name suggests, they are amylases, so they work on the starch molecules, and generally the starch molecules are converted to maltose. And lysozymes, they are active against the bacterial infection, so they will act against the bacteria if any present in food. The next enzymes or set of enzymes are the gastric enzymes, where you have pepsin, renin, and lipase. So, pepsin is generally responsible for cleaving the proteins into peptones and proteases. Then we have renin. This is present in the infants generally, and they are again working on the protein digestion. Lipases, as the name suggests, they are secreted for the digestion of the lipids. So they convert fats to the their simple units like mono or diglycerides. Yeah. So here you can see. So uh, pepsinogen is what is synthesized by the gastric gland. Remember, and this pepsinogen gets converted to pepsin when there is extra production, when there is HCl. So HCl will also be produced by gastric glands, by the parietal glands. So these parietal glands will secrete HCl. These gastric glands will secrete pepsinogen in its inactive form. HCl will activate this pepsinogen by cleaving of this part, because of which it will convert to pepsin, and then they will act on the proteins to digest the proteins into peptides. You see how this again, this is another checkpoint in the digestive system or in general in the body, where you don't have excess of something that can go and keep targeting every protein or other, that will actually lead to a non-balance or disbalance of the hemostasis. For the pancreatic juices, as I told you, that there will be multiple type of enzymes. For example, trypsinogen, we've already discussed this. Then you have chymotrypsinogen. This also we have discussed. We haven't discussed procarboxypeptidases. These are the inactive form of carboxypeptidases, again, which act on the proteins. Then, other than that, you have amylases, lipases, nucleases. As the name suggests, they act on polysaccharides, lipids, and nucleic acids respectively. And you have cholesterol esterases, which are very specific to work on the esters, cholesterol esters. Moving on, in the intestines, you have these disaccharidases, dipeptidases, lactases, nucleosidases, and lipases. Again, it ranges to a broad range of biomolecules, because intestine is the last place where the digestion takes place. So there's partial digestion in the mouth, then in the stomach, and ultimately in the intestine. Then in the intestine, you have this different kind of enzyme that act on different kind of biomolecules. It even works on the disaccharides, which are the products from the digestion of complex sugars. So to summarize all of these here, you can start from the oral cavity. You have the salivary amylase there, which are involved in the carbohydrate digestion. So there the polysaccharides or the disaccharides, they are converted into smaller polysaccharides or maltose or the simpler units. And there is no enzyme for the protein digestion or nucleic acid or fat digestion in the buccal cavity or the esophagus. In the stomach, you have partial digestion of proteins, where you have the pepsin and other enzymes related, which convert the protein into small peptides. Then, as I mentioned, in the intestine, you have the majority of digestion. So you can start with first the lumen of the intestine, the small intestine, where you have carbohydrate digestion because of the pancreatic amylases. Then for the proteins, you have trypsin, chymotrypsin, and the carboxypeptidases, which ultimately lead to the cleaving of the protein into amino acids. You also have nucleases, where the DNA RNA gets cleaved into nucleotides, and the fat globules are also digested via the presence of bile juices and lipases. And the remaining of these products are then further digested in the epithelium of the brush border cells of the small intestine, where you have the disaccharidases, all these different kind of enzymes that we just read in the previous slide. They are the ultimate products of digestion are monosaccharide, amino acids, and the nitrogen is basically so the unit component of these complex biomolecules. And once the digestion process completes, that the last step for the digestion process is for absorption. So in absorption, you have the simple nutrient molecules, which are absorbed via the GI tract or the gastrointestinal tract. Along with nutrients, you also have water that is being absorbed. It happens in stomach, in the large intestine, along with some minerals and vitamins getting absorbed. And maximum of this happens in the small intestine because of the presence of the villi-like structure. However, there are some components which are specifically absorbed at specific region. For example, iron in specifically duodenum, and when vitamin B12, which is absorbed in specifically in the ileum. So duodenum, ileum, and jejunum are all part of small intestine. And then what happens after being absorbed from the small intestine into the bloodstream? These nutrient molecules are transported to different parts of the body for either storage or further chemical modification or energy requirement. So these, we'll see how these different biomolecule move across the small intestine. So you have these fatty acids. So they move into the mucosal cell of the small intestine by simple diffusion, just by gradient of their concentration. Water molecules also move according to the concentration gradient, but the process is called osmosis. You have, if you have fructose, then you will have these something called facilitated diffusion, where even though it will move again in the according to concentration gradient, but it will not happen on its own. It will require a facilitator, basically a helper transporter, which will transport this from the outside region to the inside region. So for example, here you will have something called transporter, blue transporter, which will bind to the fructose at one end and then push it inside the cell. And for the amino acids, you have the active transport. Active transport is transport against the concentration gradient. So generally, if we are to saying that the movement is according to concentration gradient, what we mean is that it will move from a region of high concentration to low concentration to lead to something called equilibrium. But in amino acids, it moves from a region of low concentration to a region of high concentration. So that is via active transport, and that requires energy, involvement of energy. And how does this absorption takes place? This absorption takes place at the epithelial tissue lining of the small intestine, and there you have these several wrinkles which are covered by these small structures like villi. And what happens, all these villi allow this movement of the nutrients from the outside into the cell, and you can see presence of this villi increases the surface area for absorption, because each of these villas will have a network of blood capillaries. You can see each of these villas here has these blood capillaries connected to them. So these are the brush border cells. All of these micro, so take one microvilli, enlarge it. All of these are layered by these brush border cells and then these capillaries. And these capillaries are what will, where the food will, nutrients will finally be transported to. So here the nutrients will, so with the thing that we saw here, the lumen is basically this lumen that we are talking about inside the cell. And in the blood capillaries, the molecules get absorbed in the form of, let's say, simple sugar, amino acid, glycerol, salts, and water soluble vitamins. And you also have limbs. These limbs, which are like lacteals. So where the limbs are present, they are called lacteals. So these absorb the fatty acid. So for the fatty acid absorption, you have lymph, and for all the other water soluble components, you have the blood, and they then transport the required nutrient to the required destination. And that is happening via this, as I have mentioned already. So this is how the movement takes place between the intestine lumen and then the interstitial fluid that is moving to the blood. And this is for the fats, which will move for finally into the lymph. So, to conclude today's session, there are some set of questions that we will answer, and then we can wrap up this session. So, the first is the enzyme which are present in the pancreatic juices are which of the following combinations. Next is which of the following is synthesized and stored in the liver cells. Next, the slit-like space between cheeks and teeth is referred to what? We, even though we haven't discussed this in today's session, we have discussed this previously. And the last is the involuntary muscle contraction in the esophagus is called what? The first question answer is the juice enzymes are present in pancreatic juices are trypsinogen, lipase, amylase, and procarboxypeptidases. In the liver cell, you have the synthesis and the storage of glucose as glycogen. The slit-like space is called vestibule, and the involuntary muscles of this esophagus, which is peristalsis, it is controlled by medulla oblongata. Next, enterokinase helps in the conversion of which of these biomolecule. The walls of the small intestine consist of finger-like projections that we have discussed. What is the enzyme that breaks down lactose? Again, you should find this enzyme name in. So, if you want to guess the name of an enzyme, you can look at the name and try to guess the product that it works on, or look at the product and then try to see the name of the enzyme that it can possibly be acted upon by. And the last is the movement of chloride ion during co-transport of glucose into the intestinal epithelium during absorption is by a movement of what or via which movement? So enterokinase was for trypsinogen to trypsin. So remember we had spoken about the activation of trypsinogen to trypsin that is taking place in via the enterokinase. Lactose, as the name suggests, is being acted upon by lactase. Villi increases the surface area in the small intestine and they are involved in the absorption. And chloride ions, which are co-transported with the glucose, they also move by diffusion. And the last set of questions are pancreatic juice is secreted or stimulated by the release of which of these molecules? In human, lacteals are found. We have discussed where we find lacteals. So where are they found? Cholesterol esterase and phospholipase enzyme are produced by which of these organ or part of the body? And we have discussed cholesterol esterase, so you should be able to guess it. And what stimulates the production of gastric juice in the stomach? So pancreatic juice is stimulated by the release of secretin and cholecystokinin. These are hormones that increase the secretion of pancreatic juices. Um, Lacteals are found in ileum, and these were part of the lymphs. Where lymphs are found in the lacteals, so for the absorption or reabsorption. And cholesterol esterase, as we have discussed, they are produced by pancreas. And again, gastric juice, they are stimulated by gastrin. So gastrin is again a hormone which acts for increasing in the production of gastric juices. So that wraps up today's session. And we are left with few more topics to be discussed that I will be discussing in the coming lecture. Thank you.